Hello, this is Ashwati, Assistant Professor, Department of Psychology, Sri Krishna Arts and Science College. And today's lecture is about impulse transmission. As you know that neurons are the building block of nervous system. The major function of the neuron is impulse transmission. We'll be discussing about it in this lecture. And in this lecture specifically, we'll be talking about the resting membrane potential of. Yeah, in impulse transmission, we can see that the major topics included under the impulse transmission as neuron resting potential, action potential, radon potential, excitatory postsynaptic potential, and inhibitory postsynaptic potential. And also we'll be discussing about a few neurotransmitters. Yes. As you can see here, this is the basic structure of a neuron or the building block of nervous system. So just like any other cell, the neuron cell body will also contain different organelles like endoplasmic reticulum, mitochondria, lysosome, ribosome. But here we will be focusing on the major parts of the neuron. So the cell body will contain a nucleus which control the entire neuron. It will contain genetic material, and then from the cell body, you can see multiple tree-like projections, shorter projections, and these are called as dendrites. And dendrites are for receiving signals from the other cells. And the other cell can be a muscle cell or another neuron or a gland. Okay, so information will be received by the dendrite. And from the cell body, you can see a longer single projection, and this is called as an axon. And axons are responsible for impulse transmission, and the impulse will be generated at the axon hillock, the portion between the cell body and the axon. It is the starting point of the axon, which is called as axon hillock. And then in the axon you can see there are insulations, and the insulations are called myelin sheet, and this is for increasing the speed of the signals, and it is formed by the repeated rotation of the Schwann cells or an oligodendrocyte. In a central nervous system it is oligodendrocyte, and in a peripheral nervous system it is Schwann cells. So basically the Schwann cells will rotate around around the axon and forming a thick lipid layer, and this layer sheet is called as a myelin sheet. It provides insulation for the axon so that the conduction can be faster. So you can see here the axon is divided into certain branches, and this is called as axon terminal. So there are portions where the myelin sheath is absent, and the portions are called as nodes of Frank. So in a myelinated neuron, the impulses or the action potential will only generate at the nodes of Frank, making the conduction faster. So the impulses will jump from a nodes of Frank to another one, so that the impulse transmission will be faster in a myelinated neuron. So yeah, the major function of neuron, as I said, is neural communication. That is the way the neuron communicate with each other. That is what we call as the impulse transmission. And the information from the neuron to the adjacent one will be transferred only in the form of electrical impulses, because neuron can't understand any other language. So whatever the stimuli are, which are from the physical environment, which can be from the external environment, from like a sensory stimuli, and something from the internal environment of the person. It can be the stimuli can be from either the external or internal environment. Whatever the stimuli is, whatever it is, it should be transferred into an electrical impulse so that it can be transferred or it can be transmitted along the nervous system. So, and this, the process is called as transduction, the process of changing the external stimuli into an electrical impulse. The process is called as transduction. Yeah, so resting membrane potential, the terminology itself gives the idea about what is our resting membrane potential. It is basically the potential or the electrical difference of a neuron, that is the neuron membrane or the neuron at rest. We can say that it is the potential of the cell at rest. Simply, it is the potential of the cell at rest. And this is the value of minus 70 millivolt. And as you know that electrical difference means because there'll be a positive charge and a negative charge in a particular neuron, specifically the outside of the neuron is positively charged and the inside of the neuron is negatively charged, and this is said to be a polarized neuron. The process is called as polarization, that is outside of the neuron is positively charged and inside is negatively charged. In a resting neuron, this is the charge distribution, and this charge distribution is maintained by the ionic gradient. Yes. Now just take a close look at the plasma membrane. We can see that there are several ions are distributed, and you can see that outside of the cell the ions which are concentrated are majorly sodium and chlorine, and inside you can see that uh, potassium and anion concentration is higher. So at the rest, in a resting neuron, the concentration of sodium and chlorine ion will be higher on the outside of the neuron, and the concentration of potassium and anion will be higher in the inside of the neuron. And now we'll be in doubt, why is this happening? Why this charge distribution, or why this ionic gradient is, how is this forming? Yeah, we can move on to the next slide. So yeah, in a plasma membrane, you can see that there are different gates in it. That is, plasma membrane is made up of a lipoprotein, and in that we can see that there are several gates. That in this picture you can see green, blue, and yellow type of gates. And yeah, here these all gates are for specific functions. Yes, in a plasma membrane, there are a variety of voltage gated channels, and here specifically there are sodium gated channels, potassium gated channels, and then additionally there are sodium potassium pump. So sodium gated channels are a type of voltage gated channels which operates on the voltage difference, and which will allow the passage of only sodium ions. And then there is potassium gated channels which will only allow the passage of potassium ions in and out of the cell. And then there is sodium potassium pump which operates on ATP, that is adenosine triphosphate, which is actually made up by the mitochondria, which is the energy, which is actually the energy, and it is an active transport. Whenever we use energy, it is called as an active transport. And this sodium potassium pump actively pumps three sodium outside and two potassium inside. Yes, and then in the resting potential, we can see that the sodium um, is the sodium membrane and sodium gated channels are actually closed, and the potassium channels are also closed, so that the sodium remains outside and the potassium remains inside. And the only thing that's working is the sodium potassium pump. You can see in the picture that it is pumping out three sodium and pumping in two potassium. Yes, and this 3:2 ratio is very crucial for maintaining the electrical differences. And now you'll be having that doubt that on the inside also there is positive and negative charge. That is the anions and potassium ions. And outside also there is positive and negative ions, that is chlorine ions and sodium ions. But still inside is more negative. And this is because of the sodium potassium pump. There are 3 to 2 ratio of pumping ions, that is 2 two potassium inside and 3 sodium outside, and then both the sodium and potassium gates are also closed. And moreover, there are anions which are inside of the cell membrane, that is inside of the cell, and they are highly negatively charged, and also they can't move. There are no gated channels for the passage of anions. That's what make the resting membrane a lot more negative inside and positive, comparatively positive outside. So as you can see here, this is the structure of the neuron and how the concentration or the concentration lines or the distribution lines are in it. You can see that. And yeah, we can move on to the summary. So in this lecture, we have discussed about the resting membrane potential, which is the electrical potential difference when the cell is not firing or when it is at complete rest. And then outside of the neuron is positively charged and inside is negatively charged. Outside, the concentration of ions like sodium and chlorine is higher, and inside, potassium and anion is higher. And how this is maintained because of uh, voltage gated channels which only operates under voltage difference. Okay, so the sodium gates are closed and the potassium gates are also closed, and the sodium potassium pump maintain the ionic balance by pumping out 3 sodium outside of the cell, and 2 sodium, sorry, 2 potassium inside of the cell. This is how the resting membrane potential is maintained, and it is of the magnitude minus 17 milliwatt. So these are the few key terms that you should remember: polarization, the resting membrane potential, model gated channels, sodium potassium pump, and plasma membrane. I hope you understood the lectures, and if you have any doubts, just comment down, and we'll be back with another interesting lecture. So till then, thank you so much.